Welcome to today's video where we'll be diving into the realm of epigenetic regulation and gene expression. Epigenetic regulation of gene expression is a fascinating area of study in biology that has transformed our understanding of how genes function. At its core, epigenetics refers to changes in gene activity that do not involve alterations to the underlying DNA sequence. Instead, these changes are brought about by modifications to DNA itself or the proteins known as histones, around which DNA is wrapped. These modifications can affect how genes are turned on or off, influencing a wide range of biological processes, including development, age, and disease. To understand epigenetic regulation, it is important to first grasp the basics of gene expression. Gene expression is a process by which the information in a gene is used to produce a functional product, typically a protein. This process is tightly controlled to ensure that the right genes are expressed in the right cells at the right times. While much of this control is exerted at a level of transcription, the process by which DNA is copied into RNA, epigenetic mechanisms, as an additional layer of regulation. One of the most well-known forms of epigenetic modification in DNA is DNA methylation. DNA methylation involves the addition of methyl groups to the DNA molecule, typically at cytosine bases that are followed by guanine bases, in configuration known as CpG sites. When methyl groups are added to these CpG sites, the associated genes are often silenced or turned off. This is because methylation can prevent transcription factors, the proteins that initiate gene expression, from binding to the DNA. Methylation can also attract proteins that compact the DNA, making it less accessible for transcription. Another key player in epigenetic regulation is histone modification. DNA is wrapped around histones, forming a structure known as chromatin. The way in which DNA is wrapped around histones can be influenced whether genes are accessible for transcription. Histones can undergo various chemical modifications, such as acetylation, methylation, and phosphorylating, and ubiquitation. For instance, acetylation of histones typically relaxes the chromatin structure, making the DNA more accessible for transcription factors and promoting gene expression. Conversely, decylation of histones can lead to more compact chromatin structures and DNA repression. The epigenetic modifications are not just static marks, they are dynamically regulated by a range of enzymes. For example, DNA methyltransferase are enzymes that add methyl groups to DNA, while demethyltransferase removes these groups. Similarly, histones acetyltransferase adds acetyl groups to histones, and histone desynthetase removes them. The balance between these opposing activities determines the epigenetic state of a gene, influencing whether it is active or silent. Epigenetic regulation is crucial during development, as an organism develops from a single fertilized egg into a complex multicellular being. Cells must become specialized into different types, such as muscle cells, neurons, or skin cells. This process, known as differentiation, requires a selective activation of repression of specific genes in different cells. Epigenetic modifications play a central role in the process by ensuring that the right genes are expressed in the right cells. Once established, these epigenetic marks can be stably maintained through many rounds of cell division, allowing differentiated cells to maintain their identity. Interestingly, while epigenetic marks are generally stable, they can also be influenced by environmental factors. These include diet, stress, and exposure to toxins that all lead to changes in epigenetic marks, which in turn can affect gene expression. For example, studies have shown that the maternal diet during pregnancy can influence the DNA methylation patterns of the offspring, potentially affecting their health and development. Similarly, exposure to certain chemicals can lead to epigenetic changes that increase the risk of diseases like cancer. Epigenetics is also closely linked to disease. Abnormalities in DNA methylation patterns have been observed in various cancers. Typically, tumor suppressor genes, which normally help prevent uncontrolled cell growth, are found to be hypermethylated and silenced in cancer cells. On the other hand, oncogenes, which promote cell division, may become hypermethylated and overactive. Understanding these epigenetic changes is not only crucial for understanding the development of cancer, but also for development of new treatments. Deacetylase inhibitors are being explored as potential cancer therapies. Beyond cancer, epigenetic mechanisms have been implicated in a wide range of other diseases, including neurological disorders, cardiovascular disease, and metabolic conditions. For example, in degenerative diseases like Alzheimer's, abnormal histone modifications have been observed in affected brain regions, potentially contributing to the disease process. Similarly, in cardiovascular disease, changes in DNA methylation patterns have been linked to the development of atherosclerosis, a condition characterized by the buildup of plaques in arteries. One of the most intriguing aspects of epigenetics is the potential to explain phenomena that may not easily be accounted for by classical genetics. For instance, identical twins who share the same DNA can exhibit differences in susceptibility to disease, behaviors, and other traits. These differences may be partly due to epigenetic modifications that accumulate over time in response to different environmental exposures and life experiences. Moreover, epigenetic changes can sometimes be passed from one generation to the next, a process known as transgenerational epigenetic inheritance. The potential applications for epigenetic research extends beyond medicine and into areas such as agriculture, environmental science, and even evolutionary biology. In agriculture, understanding epigenetic regulation can lead to development of crops that are more resilient to the environment stresses like drought and disease. By manipulating the epigenetic marks that regulate genes involved in stress responses, scientists can potentially create plants that are better adapted to the conditions of changing climate. This approach, sometimes referred to as epigenetic breeding, could complement traditional genetic breeding methods, offering a new way to enhance crop performance without altering the underlying DNA. In environmental science, epigenetics offers a powerful tool for understanding how organisms respond to environmental changes. For instance, researchers are investigating how pollution and other environmental factors can cause epigenetic changes in wildlife, potentially leading to altered behaviors, reproductive patterns, or even the emergence of new traits. These studies could provide insights into how species adapt to changing environments and help identify early warning signs of ecological stress. Epigenetics also has a profound implication for understanding of evolution. Traditional evolutionary theory focuses on changes in DNA sequences as the primary driver of evolution. However, epigenetic changes, which can occur more rapidly than genetic mutations, may also play a role in how species evolve. For example, epigenetic modifications could allow organisms to quickly adapt to new environments by altering gene expression patterns without waiting for beneficial mutations to arise. Over time, some of these epigenetic changes may become stabilized or even influence the genetic code itself, contributing to long-term evolutionary change. One of the most exciting areas for epigenetic research is the potential to inform personalized medicine. These epigenetic modifications can vary greatly between individuals. Understanding a person's unique epigenetic profile can lead to more personalized and effective treatment for diseases. For instance, in cancer therapy, analyzing the epigenetic landscape of a tumor can help identify which genes are abnormally silenced or activated and guide the selection of targeted therapies that are most likely to be effective for that particular patient. Epigenetics also holds promise for the field of regenerative medicine. By reprogramming the epigenetic marks of adult cells, scientists can potentially revert them to a pluripotent state, similar to embryonic stem cells, which can then be used to generate any cells in the body. This approach can lead to a new treatment for a range of conditions, from spinal cord injuries to degenerative diseases like Parkinson's disease. While still in its early stages, research in this area is advancing rapidly and can revolutionize the way we approach tissue repair and regeneration. As we continue to explore the complexities of epigenetic regulation, ethical considerations will also become increasingly important. For example, as epigenetic therapies become more widespread, questions will arise about the potential long-term effects of altering epigenetic marks, especially if these changes can be inherited by future generations. Additionally, as our ability to modify these epigenome improves, the possibility of epigenetic enhancement can lead to debates about fairness and ethics of using these technologies, to alter traits like intelligence, physical ability, or even lifespan. Another area of concern is the potential for epigenetic changes to accelerate health disparities. Since environmental factors such as diet, stress, and exposure to pollutants can influence the epigenome, populations that are already vulnerable due to socioeconomic conditions, they might be a greater risk for epigenetically mediated diseases. This raises important questions about how to address these disparities and ensure that the benefits of epigenetic research are accessible to all. Despite these challenges, the potential for benefits of epigenetic research are immense. As we learn about how mechanisms regulate gene expression, we are more likely to discover new ways to prevent, diagnose, and treat a wide range of diseases. Furthermore, the insights gained from studying epigenetics will continue to deepen our understanding of fundamental biological processes, from development to aging to evolution. In conclusion, the field of epigenetics represents a powerful and dynamic area of research that is reshaping our understanding of biology. Through the study of how gene expression is regulated by modifications to DNA and histones, scientists are uncovering new layers of complexity in the regulation of life's most fundamental processes. While we have already learned that a great deal about the role of epigenetics in health and disease, the field may still in its infancy, and many exciting new discoveries still lie ahead. As we continue to explore the intricacies of the epigenome, the knowledge we gain will undoubtedly have a far-reaching application, not only for biology and medicine, but for society as a whole. So, if you are excited about the future of epigenetics as I am, join us in our next video as we will unveil the miraculous new discoveries happening in the world of biology today. Stay tuned.